Hi everyone, thank you for watching our video on population-specific genetic variation in large sequencing datasets. My name is Jeroen van Rooij. I'm a PhD student at the Erasmus Medical Center in Rotterdam in the Netherlands, and I'm making this presentation to accompany our short report in the European Journal of Human Genetics. In this short presentation, I would like to show you some of the work that we did and the conclusions that we could draw from it. Our work is focused in the Rotterdam Study, which is a single-center population-based cohort study in Rotterdam in the Netherlands. The study started in 1990, nearly 30 years ago, enrolling participants of 55 years or older to investigate traits and diseases that develop during life, so age-related diseases or traits. And as you can see in this image, those individuals every 4 or 5 years would come to our research center and get screened for all kind of variables. So we can see the individuals that develop an age-related disease or trait and what their measurements were before and after that event. We also included additional participants in that same population-based cohort study at later intervals in the study, and there are specific articles where more details about this are described. Every time one of those participants comes to our study, our research center, we do a lot of measurements, about 1,500 determinations per subject, and for the oldest participants in our study, we are at the 6th follow-up, if I'm not mistaken. We monitor, in addition to all of those measured variables, also their medication use through the general practitioners and the pharmacy. And on the left here we see an overview of a number of those age-related diseases or traits that have developed. This is a slightly outdated number, these have increased by now, but we see many individuals, many participants that develop cardiovascular or neurodegenerative or other types of age-related illnesses, and these are being studied by the various research groups within the Rotterdam Study. One of the research topics that is studied a lot in the Rotterdam Study is genetics. In the past, using GWAS technology for all of the individuals in our study, a total of about 15,000, we were able to measure the absence or presence of genetic variations in relationship to these various traits, which has been very helpful. And to continue that line of research, we have now also done whole exome sequencing on 3,000 individuals from that baseline cohort, so the oldest participant group in our study. For this, we use the SECAP version 2 with 44 MBs of target. We do 2 times 100 base pair sequencing on the Illumina HiSeq, and then use a relatively standard BWA GATK pipeline for all of the processing, using the GATK haplotype caller with multi-sample calling through the GVCS. And there's more details about this in the supplementary material of our short report. Going a little bit more into detail here, out of the 8,000 participants from the study, we selected those that had phenotypic information available, as well as informed consent and DNA. We ended up sequencing just short of 3,000 individuals. We then did a number of technical QCing procedures to remove samples that were not sequenced very well, or where, for example, the concordance to the previously run arrays was not sufficient, but also biological QC to remove outliers on the number of variants that we find, for example, but also the number of heterozygous or TITV variants that we observe. Resulting in a final data set in this freeze number 2, which we call ERA62, Rotterdam Study Exome Sequencing Freeze 2, with 2,628 individuals. And in the future, more freezes will be made and released, but this is the one that we described in our short report, and that's being used for various research purposes at this moment. To give you an idea of some of the work that we do with this specific data set, we have some examples on the right. Doing the relatively standard case control studies of the various diseases and traits that I mentioned, looking at rare mutations, also on gene burden tests, but and finally also fine mapping some of the previous GWAS loci that were observed. And these are areas where sequencing has added value over the GWAS data that we already had available, although these two type of data sets complement each other in some of this work. We also do replication studies with other cohort studies like the CHARGE consortium, but also trait-specific projects like the European Alzheimer's disease exome sequencing project and others like this. We look at loss of function variations in specific genes, genes that are clinically relevant, for example, and then go back in carriers of these mutations to see if they developed some kind of phenotype, or if some of the clinical information indicates a deviating phenotype or trait in these individuals, and try to assess the impact of specific variants on these genes in these individuals. Another thing that we do is look at population genetics, where we compare the variants that we find in our data set with those found in other similar types of efforts. And we have an additional slide on that and more information in the short report, where we compared the variants, the coding variants in our data set with those in various other population databases that are commonly used for either filtering purposes or to function as some kind of reference population of healthy individuals. And we see that many of the variants that we observe are also observed in these various populations, but there are also many variants that are not observed in these populations. And when we compare them to the various databases, we see, for example, an overlap here with the Genome of the Netherlands, another Dutch reference data set, the United Kingdom, UK 10K project, the 1000 Genomes, the ESP, the Exact Aggregation Consortium, and even if we combine all of those data sets into a very large sample set of almost 74,000 individuals, we still see that 120,000 of all of the variants that we find in the coding exons are only observed in our own data set. And it's probably not a surprise that most of these are very rare variations that occur just once or twice or maybe 5 times in our data set. But it does show that even though all these large data sets are available, the total sample size of this combined data set is almost 30 times that of our own. We still find new variations, and that was true for each of these data sets. They contain variants that are not found in any of the other databases, which shows us that there are differences based on ethnicity, for example, but also on the type of capturing that was used, or the type of sequencing that was used, or the underlying populations that are studied. And we think that using multiple data sets when you are looking for a reference set or a control set, that will give you added value over any one of these, even the largest one in Exome in this case. To summarize and conclude that, we see that if we use population datasets as controls, there are a lot of differences, and we need to evaluate how the study population of that database is in relationship to our own. In example, some of these databases include cases, so if you're looking for a healthy control population, you need to make sure that your type of case isn't included in the study design of that particular database, but also the ethnicity of that data set is similar to the one you have, or else you will find very different frequencies or presence or absence of variations. Also, if we use multiple data sets, we see that we get a better resolution, so we can remove more variants that are present in either one of those reference populations, even if there's a large difference in size, but just because these databases are different both biologically as well as technically, so using multiple is always better, especially if you have a local database in addition to a large aggregation database like Exome. And finally, the database that we generated, named RSX2, is available as one of those healthy control population databases for filtering of population genetics purposes. And we think this is particularly useful when you have a European or even Dutch or Caucasian data set that you want to study, because that matches very well to our control population. That leaves me to acknowledge all of the individuals who helped to make this possible. If you have any questions or you want more detail, do not hesitate to contact one of the authors about it. Thank you.